And we think that there's still a lot to learn about how innovation really occurs out in different areas. We were started 25 years ago in San Diego, leaders at the University of California, San Diego, where we came from, and leaders in the San Diego business community did not feel like there was enough development of all the great research that was coming out of UCSD, and they worked to create Connect with the express purpose of moving research and discoveries into commercial products, companies that could grow jobs and grow the San Diego economy. So since then, we have helped over 2,000 companies get started over that 25-year span. They contribute to the economy each year, $150 million in investment per year, double the workforce. And just recently we opened our DC office with the purpose of trying to tell that story of the San Diego Innovation Hub to DC and helping DC learn about innovation through real-life experiences of what we've discovered in San Diego. And so that's the purpose of this briefing, is not to have me tell you how it works, but to have you all hear firsthand from the innovators, the scientists, the venture investor, the leader of the Proof of Concept Center firsthand, so that you can hear how it's really like. So over the last 25 years, what's developed in San Diego is a core of research institutions that produce various research, and we partner with our great university systems. We will be talking about the Von Liebig Center, who is the Proof of Concept Center that helps translate that research into companies and jobs. We have various clusters that have developed: defense and security, wireless software, life sciences, and the company we'll be featuring today, Flamagen. They'll be telling their story. Energy and environment. Sports innovators is our most recent growing hub. People have realized they can make a better surfboard, a better helmet, better apparel, and it's really exciting to see these folks that like to play a lot realize they can make money by making a better surfboard. What the other thing we do is we help other trade associations in San Diego get started as they focus on their individual industry. Last year we were fortunate enough to win the Innovation Award from the Economic Development Administration, and we have some folks in the audience from there, and we appreciate them coming out. And this is one of the key pieces that I wanted to mention. You know, there's so much buzz in Washington, D.C. about creating jobs. We really think it's important that you focus on tech sector jobs, because those jobs pay better wages and tend to be a better kind of job. In San Diego, what we found is that the tech sector is only 5% of employers, only 10% of all jobs, but those 10% make 25% of all the wages in San Diego County, but those wages are 95% higher than the average wage. So we would say that certainly we need gas stations, cleaners, restaurants, but if you want the high wage jobs, then the administration and Congress should really be focusing on how do we drive tech sector growth, what are the impediments that prevent tech companies from growing, and hopefully today you'll hear some answers. So I want to, without further delay, introduce Rosabelle, who will come and talk about the Von Liebig Center and all that they've been doing.
Good morning. The mission of the Von Liebig Center is to accelerate the transfer of university discoveries to the private sector and to prepare our science and engineering students for the entrepreneurial workplace. By doing that, we bring industry and university closer. We started 10 years ago as a proof of concept center founded at UC San Diego with a $10 million gift from the Von Liebig Foundation. In 2007, that funding was spent down. So now we support our operations by a combination of funding from the federal government, the Department of Energy, TATRIC, private foundations, and the university support. We provide a combination of business mentoring from seasoned entrepreneurs, pre-venture grants that pay for the graduate students to do the work in the lab, and graduate level education for our science and engineering students. We are also a forum that brings together innovators, entrepreneurs, mentors, and students in a very supportive environment. As I'll show you in a minute, this is a platform that can serve multiple institutions in a geographical region and also can be replicated. We are located really at the transition. We work with faculty at the transition of research in the laboratory when they start the actual commercial development. Today you will hear from Professor Schmidt-Schonbein and Mr. John Roddenreis. They are the founders of Inflammagen. We started helping Professor Professor Schonbein in 2002 through 2007, and we provided $150,000 in GAC funding plus more than $50,000 in business mentoring. And today you'll hear what is their journey through the valley of death. So we are really at that transition, the beginning of that valley of death phase. Proof of concept centers are important, and proof of concept in general is important because you have to go through this stage in order to demonstrate market viability of technologies. And universities are a great place to accelerate innovation. But as you all know, moving IP out of the university is not easy. Why? Because the technology found in the research laboratory is not ready for the marketplace. There's no demonstration that it works outside the lab. The market is sometimes unknown, and the business model is not clear. There's no management involved. We have a professor with their graduate students interested in seeing their invention getting to the private sector, to the public. There is a culture gap between research and business. There's a different sense of urgency, motivation, and priorities. So how is our model helping to accelerate translation? There are 4 key elements. One is the pre-venture grants, $50,000 to $100,000 that pay for the graduate student or the postdoctoral researcher to conduct proof of concept studies in the laboratory, guided by a business advisor and their faculty advisor. This funding is released upon milestone completion and is highly leveraged because it's done inside the university infrastructure. The advisors are seasoned entrepreneurs, business executives that are experienced in product development and commercialization. They guide the researchers throughout the commercialization process, from beginning to end. And they bring their strong connections to the regions, from the region's innovation ecosystem. Our education programming, we've been doing this for 10 years. More than 1,000 students in engineering and science have gone through our programs, entrepreneurism. Our goal is not necessarily to create entrepreneurs, but to train our students to think entrepreneurially and to understand how technology companies innovate and compete globally. We have a very well-characterized process for filtering and sorting what the projects and the teams deserve the funding. So we focus limited resources on those technologies that will have the greatest impact. Our process lasts about a year and a half. From the moment that the researcher and his or her students come in and apply for services, we assign them a mentor. And this mentor works with them throughout the process for a year and a half and more sometimes. And upon graduation, they help these technologies be shaped into products or services that then can leave the university in the form of a company. Those companies then are ready now to apply for SBIR funding, engage with venture capital, angel investment, or join an incubator or an accelerator. We also work very closely with MBA students to help put together business plans for these technologies. Since 2007, we started expanding the program and offer it to other universities in the region. So we created what we call technology acceleration programs, and we have done 5 of these programs in areas of regional interest: clean technology, wireless health, renewable energy, and innovations that reduce the cost of delivering healthcare. We have worked with 150 researchers from 13 universities and institutes in Southern California that have participated in this process. And we have already funded more than $1 million in grants. So we have invested in these 10 years $4.6 million in grants to 82 projects, funded close to 100 graduate students and postdoctoral researchers to do proof of concept studies. We have helped start 32 companies, 200 high-value jobs, and we have worked with more than 300 technologies and have more than 25 projects in the pipeline. So what have we learned? We have many lessons to share with you, but I thought that these were some of the most, in a way, obvious but also relevant. There is a huge gap between an idea and a product, and bridging that gap is not done by the researchers alone. You need the entrepreneur. And there's a quote that says that innovation without entrepreneurs goes nowhere, but you have to bring them together. Researchers need business-oriented guidance throughout the technology validation, and that guidance needs to be impartial and honest. A well-structured process that selects the best candidates and ties grant payments to achievement is critical because it keeps the researchers focused. The pre-OC process should focus also, we should always focus and put our efforts in the role of the graduate student and the postdoctoral researchers. They are the ones that leave the university and join the companies or start the companies. The faculty do not. We should also expand the role of the Proof of Concept Center and leverage these very smart and talented students to assist small and mid-sized companies, address their needs, solve their problems, and accelerate the translation of innovations from academia so they can remain competitive. Thank you.
Good morning. My name is Geert Schmidt-Röhnbein. My responsibility this morning is to tell you about the science. So I'm not a businessman, and I'm very fortunate to have a businessman who works together with me, which makes all of that possible that I'm going to tell you about. Also, I'm going to tell you a new story you haven't probably never heard in your life. So enjoy that part. It's called autodigestion and what's eating us. The word autodigestion has been used previously in science, but I recovered it and I'm using it now in a new context. All of this work is only possible because there's an entire team of people, and I had the great fortune that in addition to the funding from the Liebig Foundation, I had for decades funding, and I have from the National Institute of Health. All of that only made it possible to even think about the things that I'm now thinking about. And then, of course, it was necessary in order to translate these ideas, because I had a very strong feeling I can save lives, congressmen's lives, congresswomen's lives. I was very anxious to move that forward into a real practical application. So the story I'm going to talk about is sepsis and physiological shock. At that point, the patient is in an intensive care unit, hooked up with all kinds of pipes, and his life is on the line. The chances that this individual will survive quickly reduces as one organ after the other one fails. Kidney fails, the lung fails, and so on. And the statistics indicate that by the time you're at 3 organs that fail, chances that you leave that hospital is now down to 0. We spent many, many years, decades, thinking about this question: where does this phenomenon come from? And I would say after 2 decades of work, have come to realize there's something very special in our body. And don't think this is just an invention by Dr. Schmidt-Röhnbein. It took me many years, many experiments where I thought something else was going on, until nature pulled me by my hair to your intestine. Okay? You have an intestine because that is required to get all the energy that we as large species want to have. And if you look at an intestine, there's a very peculiar phenomenon by which after the swallowing, our food arrives in the stomach. It's acidified there, but then something very dramatically occurs. Out of your pancreas, which is this little yellow organ you see here below, comes a set of enzymes that you produce from day 1 of your life to the last day of your life that mixes with the food, and then it goes on this long ride through this intestine. And the food that you just ate, all the good steaks and so on, is there broken, at that point broken down into its molecular constituents. So polymer is broken down into individual amino acids, and those amino acids is what you are absorbing. Now, what we've been thinking, in fact, medicine in general, been thinking about this is a perfectly working process. Not so. This relies on a problem that I'll explain to you. Namely, it relies on the ability to maintain that process in your intestine. And what I'm going to tell you today, that can fail. You can always ask the question, interesting dinner conversation, if you eat an intestine or you eat a German sausage, you will eat an intestine. How come I can digest that intestine, but I don't digest my own intestine? Cannibalism in nature is widespread. Humans used to do it too. So we have the ability to digest an intestine. We don't digest our own intestine. This is a problem much older than man. The dinosaurs already had to deal with this. And nature has come up with a very clever mechanism by which it solves this problem. But like all biological processes, it's never perfect. So this is a cross-section through an intestine. This is, as you know, a long tube from here to the end of the wall. And the enzymes that are responsible for digesting are listed here, coming from your intestine. They're all in the center, in the lumen of your intestine, and they're peristaltically moved along. We never know that it actually happens. It's as important as your heart. And these cells that surround the intestine, it's called the mucosal villi, are made up of an endothelial cell that's epithelial cells similar to the epithelial cells you have on the inner part of your cheek. And they are responsible to make sure that these digestive enzymes stay in the lumen of the intestine and don't get into the wall of the intestine and start to digest your own intestine. So everything hinges on the ability of these epithelial cells. You see them here in a little magnified view. They form a very tight barrier. And under normal circumstance, food or these digestive enzymes will not enter into the wall of your intestine. That's how it's written in textbook today. But many decades of research have indicated to us that life is not so simple. And what we discovered in experimental situations, that in fact, if you take an animal and you make that, to perturb that intestine a little bit, even a young teenage animal, you start to see that these enzymes, which are visible to you here by the green fluorescence, there's very little green fluorescence in that normal intestine. And by the time we perturb that intestine slightly, the entire wall is full of digestive enzymes. And these are pretty dumb molecules. They digest, and they don't care whether this is a sausage that you just ate or it's your own intestine. And what they do is they start to digest your own intestine, and the result of that is a catastrophic event in which the normal villus structure of the intestine is now completely destroyed. The patient is now completely exposed to his own digestive enzymes. Okay? And that's one of the reasons why this shock is such a lethal procedure and an event sequence of events. So this is a cartoon of many, many publications. This is your normal intestine. There's the villus structure. These Chapmans are not able to get into the wall of the intestine. But by the time you perturb the intestine, and there are many different ways to do that, these digestive enzymes get into the wall of the intestine. They remove the inner part of the intestine, and furthermore, they generate in the intestine now breakdown products. And it turns out these breakdown products are further cytotoxic. So you're going to be hit twice. They escape into your bloodstream, and they enter into multiple organs, and that's where we end up, why we end up with what we call the multi-organ failure. So there's a very interesting set of science. I go around the world and I teach about this thing now, but it's all very new. And the immediate question that arises now is, okay, if these guys are so bad, why don't we go ahead and block these guys and prevent them? Because nature has provided us with an opportunity, and there may be a new opportunity to achieve something that's never achieved in medicine, namely to rescue a patient out of shock. And the idea is actually quite simple. What we do is this is in the case of a patient, we put down a catheter, nasal gastral tube, And ideally, we put it right at the beginning of the intestine. It's called the duodenum. That's where your digestive enzymes come in. And we inflate the balloon so fluid doesn't go backwards, and then we gradually fill up this intestine. In the patient I'm going to show you, we actually couldn't do that because we couldn't image the patient. And for that reason, the only thing that actually took place is we put the tube down here. We kept our fingers crossed that the digestive enzyme inhibitor would actually be transported along the length of the intestine. Well, we've done this type of study now. I have to say, up to this point, this was all funded by the NIH, General Medicine, and I still have an ongoing grant on General Medicine. But now, I talked to the people on the IRB and said, listen, folks, we'd like to try this. If it's my kid, I want you to try it. And they said, no, not so quick, not so quick. You need to provide us with some proofs that this actually works. And so they put us on a trip having to go through a large number of animal experiments. And that was possible because Leading Venture came forward to us and said, we're willing to fund that. They knew enough of details about it that they had more trust because they were able to talk to me than a review committee would be at the NIH. That was an important, essential element. And so we carried out a very expensive set of studies that are currently in review. This is what a normal intestine looks like. I put it slightly out of focus. I hope you're not too much offended by that. This is what an intestine looks like when it's injured in this fashion. It's a badly damaged organ, and this is what happens when you're in an intensive care unit. When we block these digestive enzymes, and the only place we do this is by putting them into the lumen intestine. We don't give any IVs or anything of that nature. This is how we typically preserve these intestines. We've shown this in multiple species. And most important, here's the bottom line, that what the IRB wants to know, if you look at the number of survivors, and this happens to be a hemorrhagic shock model, in other words, it's a reduction of the blood pressure for a certain period of time in a perfectly healthy animal. You see we have approximately 3 out of 12 animals would survive such a protocol. It's typical. It's a very lethal procedure. These animals are dead within about 5 hours. When we treat the animals the way I just described it to you, we have an 83% survival. So we have a dramatic increase. And we've shown this for many different models of shock and in many different species. There's another publication coming out very shortly. So we are anxious to move on to man. And I like to translate it. I don't want to spend now 10 years going to my scientific colleagues and try to convince them about it. I have enough experimental evidence at this particular point that I want to move forward and try this in humans. And I had an opportunity to present this idea to the premier heart transplant surgeon, Professor Wei, in Taipei. And let me move on. He had, by chance, in the evening I gave the seminar, a patient who was in septic shock. And what you see here is one of his enzyme levels, called amylase, had reached astronomical levels. And he was an individual who had already heart transplant 4 years before that, had a car accident. And at that particular point, the physicians were in a situation where they said, sorry, there's nothing else we can do. So he called me up at dinner time and said, tell us one more time, what was the concentration of the protease inhibitor you gave? We recomputed it again over the dinner table. They applied it, they put down a tube, they applied it to him. They had received consent from the family. It's not a real consent. It's a type of consent where you say there's nothing else we can do for your loved ones. And of course the parents begged them, please do something for our dad. And that's what actually took place. And this individual was treated. And to the surprise of everybody, he had this dramatic reduction within a day of his amylase levels. And they stayed down during the period of time that we blocked these digestive enzymes. And he had lots of other complications, so it took a while for his white cell count to eventually come to more normal levels. It took several months. But this individual intestine looked, on the first day he was treated, it looked like this. There's a badly dilated small intestine, massive inability to even perform peristalsis in a healthy way like it would be the case in you and me. And after approximately a 4-week period, this is how his intestine looked like. And on beginning of November, this gentleman was released from the hospital and sent back home. That will now be published as a case report within the next couple of weeks. It was just presented in a meeting in Korea. And the individuals, this trauma surgeons who look at this case say it's an unlikely individual to survive because he had a heart transplant, he was immune suppressed, he had, in addition to that, a secondary injury, and he had these very high markers for septic shock. So this is the very first indication we have that this procedure may potentially work in humans. And we are therefore, of course, literally trying on a day-by-day basis now to move this forward so that there is a possibility that one of your loved ones may potentially be saved. The final point I want to make is this: all of this is, of course, only possible because I have a wonderful crew of people. An individual who played an especially important role is David Hoyt. He's currently the executive chairman of the American College of Surgeons, and he is very strongly behind that. He's in the process of publishing another paper with me that shows the feasibility of this approach. So from a purely science point of view, we are in a stage where very few of our general colleagues have been convinced, but we know enough of the details of animal experiments, we have done hundreds of those, that we are saying at this particular stage we need to have funding to carry out clinical trials and actually prove the procedure so that it has an FDA level stamp of approval if it should work out. Thank you.
Thank you, Gerd. My name is John Rodenreis, and Gerd just gave you a very eloquent description of the science. I'm going to talk about the business end of this. And first I'm going to talk about septic shock and how big a problem it is. So we've got you here for about an hour. So in that hour, in the US, 24 patients are going to die from this. So this is not an orphan disease. It's not something that happens infrequently. And it's a world problem. There's about 1 million people a year dying in the world, over 200,000 in the US. And in 2007, US hospitals billed $38 billion for this. So it's a major issue. And pharma companies have spent billions of dollars on this, trying to figure out what causes it. And there are over 100 human studies that look at a totally different mechanism than what Gerd has discovered. So in spite of that, nobody's found something that works. There's one drug approved for this. It costs $6,400 a dose. It increases survival by 6%. So if you look at Gerd's improvement in his rats and pigs, we're talking about a 70% increase. So this is an order of magnitude better. And what Gerd does that he didn't tell you is he uses an existing approved drug, a drug that's already approved in the US, to block these enzymes. So what we're going to try to do now is move it out of his lab and get it into humans. And I'm going to talk about our experience in 2 different countries, Taipei and the US. So Roosevelt gave a great description of the Valley of Death. This really refers to the difficulty people have in taking a nascent early stage technology and getting it commercialized. And the biggest issue, which you've all read about, is it's hard raising capital, both governmental capital and private. And in fact, he's been funded by NIH and NSF, and there's talk now about cutting their budget because of the budget crisis. The same exists with private capital. But that's not the only issue with getting stuff commercialized. The longer you spend in the Valley of Death, the lower the probability is you're going to make it successful. Regulatory timelines are a big issue for us. Let's assume we got the capital. Now we've got to deal with the FDA, which is a government agency. You've all heard horror stories about how long it takes. Sometimes that's true, sometimes it's not. But they're not the only one that we had to deal with in moving through this path. We want to do this at a hospital in San Diego. So we have to go to their institutional review board, which is a board of outside people and inside people that makes sure that we don't hurt patients. Well, they've got no incentive to do anything new, because if they do something that's not standard of care and the patient dies, what happens to them? They get sued. So the IRB says, we're not going to do anything different unless it's perfect. Now, Garrett's worked on quite a few animals. Well, we've had occasions with animal committees it's taken longer than it has with IRB committees. So the regulatory timeline is a real issue for us. Another issue is the attitude of why should we take a risk? If I take a risk, I may get sued. So innovation tends to go relatively slowly in U.S. hospitals. You're from the government, you've probably seen this before. When you're dealing with a hospital, you're not dealing with one person, you're dealing with a bunch of departments. And then last, another issue is, how do I deal with somebody like Garrett? You know, I'm a business guy. I'm a greedy capitalist. He's a altruistic scientist. So how do you connect those two people? And I was fortunate enough to sit on a funding panel for von Liebig several years ago, five years ago actually. I heard Garrett talk, and when he talked about septic shock and the problems, I said, this is a real opportunity, both in terms of saving lives, but also making some money. So we spent 3 months getting to know each other, and we got the same color hair, so I said, okay, there's one stripe for you. But in talking to him, I said, you know, sometimes it's like you're talking Greek and I'm talking French. I talk EBITDA, you talk gel zomography. So what do I need to do to understand what you're talking about? He said, it's simple. You got an engineering degree. He said, pick up the molecular biology textbook and read it. So I did, 1,700 pages. You know, it's all dense. But I said, if I want to understand this, I've got to take the time and get in his shoes. So I spent 6 months, and I actually read the book. So he and I, we can talk scientifically now. If I hadn't done that, this wouldn't have worked. And one of the issues is most VCs won't do that. They'll read a business plan, they'll crunch the numbers, then they'll just say yes or no. But with this kind of early stage research, you really got to understand what's in his mind. So we talk about regulatory timelines. Timelines for devices have risen substantially since 2007. Now, we're dealing with an existing drug, which means I can use this 505(b)(2) pathway with the FDA much quicker than the regular one, which is for a new chemical entity, where you don't know what's going to happen. So my talk, I'm just going to focus on how do we get it from animal research to where we're dealing with humans. That's all I'm going to focus on. So here's the players I had to deal with at UCSD. You know, I knew Garrett pretty well, but if I wanted to license it, Garrett's here in the School of Engineering. David Hoyt, who was the clinician, is in the School of Medicine. Two different fiefdoms. You probably never heard that about the government, right? They didn't talk to each other. In addition, we had a third institution. Our biochemist was there. So here I am. You know, I got a early stage venture fund. I've got some investors. I got a board. They want to make money. So what we did, I said, okay, I want your IP. UCSD wants the money. But how do I approach them? Well, it turns out there's another player called technology transfer. If I wanted to talk about this in terms of, okay, what's this going to cost to do? I couldn't talk to him. I had to go to tech transfer. So I'm facing this large bureaucratic entity that I hadn't had experience with. Well, fortunately, in this case, von Liebig was involved. So they gave me an advisor, Steve Flame. He helped me with the science, and he helped me negotiate who these players were. Without that, this would not have happened. So we went through, von Liebig gave me some money. We got to here and we said, yeah, it works in rats, short term. But I said, look, I'm not going to invest in this until you can show me that we can treat a rat that's in shock and have them live the full 2 years. And the same with a pig. So we did some parallel pig studies and rat studies, and yeah, it worked. So we increased survival from 20% to say 80%. That's a dramatic difference. So we had two different species. So we said, hey, let's try humans. Well, doing this in humans takes a lot more money. So this was late 2008. This was what I call the perfect storm. If you remember when Lehman Brothers went down and stock market crashed. So we're faced with, where do we get money from? So Garrett says, I know. I've been funded by NIH. So we talked to NIH. They said, look, you want translational research. We do basic research. Sorry. So I said, all right, I'll go to the venture community and the angels. They said, hey, you're too early, and besides, the stock market crash has left us with no money. So I said, okay, I'll go to the pharma companies. They're the ultimate customers for this, because they're the ones that are probably going to commercialize it. They said, nope, we want human data. So we had a perfect storm here. Everybody left the market. We had this promising technique where 24 people an hour are dying in the US, and we can't move it forward. So we were able to get enough funding to keep him going. And Garrett had talked to the Navy in 2000. They said, we're not interested. This is just some crazy idea. So we went back to the Naval Medical Center, San Diego, in 2008, had a meeting with the surgeons, and they said, you know, you're probably right. So they submitted a grant, got it approved, and they started testing on pigs, because we wanted somebody outside of Garrett's lab to independently confirm this. So here, we got a successful process. Took 3 years. This is for pig testing. So about the same time, one of Garrett's colleagues at the VA, the Veterans Administration Hospital in San Diego, said, this looks like it has promise. So we talked to him and said, let's do a human test. So we took about 9 months to create a protocol. Because these protocols are, you have to dot the I's and cross the T's. We submitted it to the IRB. The IRB said, 2 months later, said, this looks like pretty good, but there's some risk here. You know, what if somebody dies and somebody sues us? So they said, you need to go to the FDA and get approval for this. I said, this is going to take a year. I've dealt with the FDA before. So we took 2 months and we submitted an IND exemption, investigational new drug exemption. Big document. And I said, well, I'll hear from them in about a year. So I said, Garrett, we're just going to put this on hold. Well, 2 days later, the FDA called and said, you really want to do this and just confirm that this is what you're going to do. And we said yes. So 6 days total, they gave us an approval. Now, I've never heard of this before. I've dealt with the FDA. So this is a case where the FDA, apparently because they saw the unmet medical need, they approved it right away. So we went back to the IRB, and they took 2 months, but they said, okay, we'll conditionally approve this. So go ahead and start bringing patients in. So a month goes by, and we get an email about 3 weeks ago from another committee that said, wait a minute, there's a HIPAA form, the wording on the HIPAA form we don't like. Even though the IRB's already approved it and we're all set to take patients, you need to change the wording. So right now, this is literally a true story. We're in limbo. We're not sure when this is going to start. Now contrast that to what happened in Taipei. They treated a patient the next day. The reason the difference is, they said, this patient's going to die. I'm going to think about the patient not getting sued, because they don't sue people in Taiwan. So if you look at those differences, it's a huge difference in terms of timing. So it wasn't so much capital at this point, it's timing. So if you look at the gating factors, in terms of academic industry, it was pretty quick. Tech transfer was quick. Animal committee was slow, they typically have been. It was tough getting both kinds of funding. The IRB was slow, but the FDA wasn't. So here's a case, I can't point my finger at them because they reacted extraordinarily well. So let's roll this back and say, what if we had done what Taipei did in the US? Well, that's a 475-day difference from then to today. I actually calculated this out. That's 280,000 deaths in the US and about 50 billion in hospital bills. Now, we would not have eliminated those, but if we had improved survival significantly, we would have cut a large amount of cost out of the US hospital system. So how can you folks help? We've got some specific requests. I think there should be point of concept centers at major universities. If you're doing research, it's very, very difficult to take that research and commercialize it without somebody like Roosevelt. So the question is, how do you fund it? Well, nobody's going to give Roosevelt more money, but there is HR 1036. You know, U.S. companies have $1.5 trillion sitting offshore in profits. They're not bringing it back because they don't want to pay 35% on it. Well, this bill says, hey, if you bring it back, invest it in innovation and create jobs, there'll be a lower tax rate. You know, our treatment, which uses an existing drug that's already approved for something that kills, 30 to 60% of the patients, is treated the same way that acne is, treatment for acne. So there should be a triage here. If it's a life-threatening disease, treat it differently than something like a cure for acne. We'd like to increase early-stage revenue. There's lots of tax changes that are being talked about right now in terms of, okay, if you invest in R&D and keep it there for 5 years, we won't charge capital gains tax on it. We could increase the Reg A limits from 5 million, which doesn't do anything, to 50. Tort reform is an issue in life sciences in the U.S. A lot of decisions are influenced by a fear of getting sued. So tort reform could be important. We did not use the SBIR. I know many of my colleagues have. It's a phenomenal program. But there's uncertainty, because with the continuing resolution, you don't know if it's going to be there 6 months from now. So let's get it firmly committed and allow VC-backed companies to participate in it. So if we do this, we're going to create a lot of jobs. If you look at the last 18 years, 42% of the net new jobs came from companies with less than 100 people. And that's the kind of innovation we're talking about. We're talking about technology-based companies that can grow rapidly. This will make a significant dent in healthcare costs. With Obamacare coming, they're going to have to solve the cost equation somehow. And we're going to save a lot of lives. So thanks for listening to me, and hopefully we can work together and compress this valley of death. I'll turn it over to Tim.
Well, there you have it. You have a real-life story of the hurdles, the barriers that prevent a startup company from getting going, and fortunately, Inflammagen has made it through the valley of death. I think that the important thing to keep in mind is there are enough market barriers that make it hard for a company to succeed anyway. Distribution, marketing, manufacturing, all those other things, it's hard to start a company. But then when you put on top of it the access to capital crunch, regulatory barriers, you make it even harder. And so our main message is just as staffers and administration folks to start thinking about what can we really do, and to understand that it's not just opening up a building and selling a product. A lot of these new technologies have a long development time, and so it's important that we think through how do we make sure there's an environment that allows capital to flow to them, and also where there are regulations. I think John's point about FDA, if you have an acute disease that can really change some of the healthcare costs and delivery to a large portion of the United States. We have obesity drugs, diabetes drugs that are in the same vein as what Inflammagen is trying to do. These should have a different, faster track. That we're not saying get rid of regulation, but let's put them at the front of the line instead of an acne drug or something of that. So those are the kinds of things we want people to start thinking about. If you have any questions, we have a few minutes. If anyone has any questions, we're happy. Right here.
I'm curious, NIH sort of done down the road in translational research with the NCAT program from Dr. Collins. I know there's been some question, a lot of people say they should stick to traditional R1 grants and not get in out of it. Obviously, I'd like to hear what your thoughts are, and I think getting that sort of translational grant structure.
Well, let me tell you, in a very specific case here, we are funded to do basic research in this area. What we're not funded for is if I come in with a grant to the NIH and says, I want to do these preclinical studies that the IRB wants to see, okay, where I have to test different chalk models and different drugs. to convince the committee that this could potentially be useful in humans. Because they would immediately say, oh, no, this is translational stuff. That should be funded by a company. So you fall in between. You're never quite right in this case. I go on the secure way as far as NIH was concerned. I just fall back on a basic science part. But it falls short in doing the type of thing that we are doing with John. And also, as John pointed out, I'm not a business person. I don't have time for this type of stuff. And therefore, I talk to clinicians. And I say, hey guys, this is an interesting idea. And they say, oh yeah, it's a wonderful, interesting idea. Why do we fund this type of thing? And so I try to involve other people in the process as well. It will take years. It has already, since the very first publication of this idea was exactly in January of 2000, 12 years, 11 years or 10 years have passed before the first patient could actually be treated. It was by this group in Taipei. In the US, no clinicians, they say, oh, this is interesting, and this is wonderful, interesting science, and we meet at meetings, I'm a member of the Shock Society and so on. But it doesn't move on, okay? And I know there are a couple of congressmen that passed away in the meantime. I know I could have saved their life.
John Murtha.
So that's been the frustration on our part. And I have enough gray hairs, I don't need to publish another paper. Okay. What I want to see is does this idea actually save the life? I see lots of young males here. You guys are at a very high risk of being involved in a trauma case. And there is no treatment for these in the moment. Did I answer your question?
Yes.
Yeah.
You might want to talk about how many patients Taipei is treated and what the survival rate is.
Well, the trauma surgeon, Professor Wei, has, of course, gained confidence in this procedure. So he now, first of all, he does surgery like all trauma, heart transplant surgeons do. He picks very carefully his patients, but he is under pressure, having to take care of cases that are higher and higher risk. People come in with worse and worse conditions. It's one of the reasons why he approached me with this whole idea. And what we therefore have decided to do is that he now picks these guys with high levels of sepsis, like this amylase value I showed you. Those patients with high levels of amylase, he will now treat. In fact, in one case, he was even able to pre-treat the individual, all on a consent basis. Okay? And out of the 13 patients last year that he treated, 11 we saved alive, 2 passed away. And in fact, we know why they passed away. We could have saved them also if we had known their condition at the time better. So that is all on a consent basis. Now you have to remember that's not a statistic, because like somebody will say the guy would have survived anyhow. So we have to yet go through a formal FDA. By the way, in Taipei, they strictly adhere to the FDA rules. That's a very important issue for them. And we need to set up now a clinical trial. In Taipei, this thing is really moving forward. I can give you some details. But in the US, the clinical trial in Taipei will be interesting for Taipei, but it's not going to be accepted in the United States. And therefore, we are now organizing this clinical trial here at UCSD. And that's the one that just takes an endless amount of time to move forward on something where I know I can save lives.
Anyone else?
Actually, I have a question for Dr. Ochoa. Is that how you pronounce it?
Ochoa.
Ochoa, okay. Yeah, I thought it was very interesting, the process that you showed that went from the actual student coming in, getting the mentor, all the way to the startup of a company. So in that process, what has been the percentage of students who have actually gone on to create a business? That's part one. And then part two, and this is based on the students that participated in the program, and part two is what has been the success rate of that, of those that have started businesses in in actually maintaining and getting through the value gap.
So, yes, thank you. If you go back 10 years ago, when they formed the center, they started focusing a lot on the faculty, about the professors, thinking that it was the professors, the entrepreneurs. But from the very beginning, our funding for the center has gone through the students to pay for the graduate student. So when a company gets formed, invariably, and we have funded 82 projects, and we have started 32 companies. Invariably, in those companies, at least there is one student involved, if not more, in that company, especially in the beginning, right? It is unusual they are the founders, like the CEOs. The CEOs are usually an entrepreneur or a CEO that is experienced, but they join the companies as CTOs or maybe as technical contributors or maybe business development people. So out of those 32 companies, I cannot give you the numbers today of how many are like UCSD or alumni from the program, but I can tell you that we have several of those companies like Ortiva Wireless, Mushroom Networks, Up to You. Each one of those companies that started with 1 or 2 people, now they have 70 employees, the other one has 45 employees, the other one has 20 employees, and many of them are UCSD employees. But we started, when you look at that graph, it's a funnel, right? We started with 300. We down selected to 80, and out of those 80, 32 came out. So it's really, we're not there to pick up winners. What we want is to give an opportunity for as many to have a chance to go through this process and validate it as much as possible inside the university. Because as you heard, it's very difficult, even outside, so we want to increase the chances.
Right. Well, I think what we're looking at today is how do we create jobs and how do we make sure that we can grow the companies that do start businesses.
So the training, that's why for us it's very important, not just, it's the mentor, but the funding, but also the educational component. Hands-on, right? Hands-on, when the funding goes to the student to do the prototyping, where we encourage them, and now more than before, to take these courses on entrepreneurs and to go and attend to workshops, seminars, get educated, get connected to the ecosystem in the region. There's a lot of help, at least in San Diego, that is free help. So get the students as much connected and as resourceful as possible, so their likelihood of success is high. Because it's really not by themselves that they're going to do it. You need to have your support network.
One more question. Have other universities, are they aware of your program, and have any decided to do a similar model?
It's interesting. Yes, they are very aware. In 2008, the Kauffman Foundation published a report called Proof of Concept Centers and identified the Von Liebig Center and the Despande Center at MIT as models for commercialization because of that integrated approach. We're not the only ones. There are about, 2 years ago, there were like 60 programs around providing gap funding. But what is unique about ours is that with very little funding and a lot of mentoring, we provide this integrated program. So after 2008, I can tell you that I have had and still have visits from all over the country and also internationally. I have programs with Korea, with Mexico, with Japan that they want to replicate the program, and also visits from all over the United States. What is interesting is that, and I agree, depending on the region, you want to adapt the program to what works for you. But I think that there's a lot of lessons learned that we have for over the 10 years that we've been doing this, that we could translate and shorten that learning curve, because otherwise you'll probably have to go through what we went through until now.
Any other questions?
I have a quick question. I've heard 2 presentations here, one in terms of the proof of concept centers and the Valley of Death, the other very specific pilot study in how this could work. Healthcare policy in Congress, by and large, about money. the cost of the procedure. And here you've identified very significant billings that could be somewhat mediated or addressed. Do you have a policy recommendation in terms of healthcare debates within the bureaucracy of HHS so that there could be a more active discussion on how these types of savings could be linked to these sorts of proven or yet soon to be proven technologies?
I have something unless you want to say something.
Why don't you start and I'll finish.
Well, I think it goes back to, I think, John's point. And, you know, we've, in San Diego, we've interacted with companies that have, you know, new game-changing obesity drugs, diabetes drugs, and they, you know, they show you these maps of their pervasive problems all across the country, very high costs because of the amount of people that are involved in it. And they are still having to plod along and struggle through that regulatory approval process. And I think if you want to talk about bending the cost curve down, you have to think of where do you get your biggest bang for your buck. And when it's those drugs that can serve the vast majority of people that are obese or have diabetes or are in shock like this, you have to look at, let's get those to the front of the line. Let's get those approved. Let's still do the trials and things, but let's get them approved. I mean, why can't we accept the type A research here in the U.S.? I mean, if they're doing studies over there and they're trying to use FDA protocols, is there really a reason why we can't use that here? You know, different agencies have done different things, like the Patent Office has a fast track program where if you pay a little more, your patent gets to the front of the line and goes fast. They've also started a green energy program where if you have a patent related to clean tech technology, you get a faster track. Well, maybe the FDA, HHS should be looking at, are there ways to take some of those fast track models and apply them, not sacrificing patient safety, but thinking through what are unnecessary burdens that possibly can be relieved. And we have to give credit. The administration has made an effort to say to their agencies, you know, we need to get you guys focused on getting rid of those unnecessary barriers. Congress is doing the same thing, saying, what are those regulatory barriers? So we're encouraged by the fact that the administration and Congress are both looking at this, and I think it needs more urgency and a faster push as to finding those and moving things forward. Did you want to add anything, John?
I'll just say that from an industry point of view, we'd be happy to participate in those discussions. What we typically do is we get FDA approval for something, then we go to CMS, and we say, will you reimburse this? And one thing the FDA and CMS have done lately is there's a parallel path now. I'm not sure how developed that is, but they're going to try to take, I think, 5 pilots and move them through both at the same time. But from an industry point of view, I'd love the opportunity to sit down with somebody and say, look, here's the cost benefit on this. If 60% of these people are going to die, and our drug might cause 2% adverse reactions, and we can save lots of money, let's do that. So I think if you reach out to industry, you'll find a positive response.
Address. All right, well, thank you all for coming out. My name is Tim Tardy-Bone. I'm here full-time, and so feel free to grab a card. I can always come up to the Hill, visit with you, or if you're in the administration, if you have any questions, I'm happy to do follow-up. And we can also get you connected to them if you have specific questions as well. Thank you for coming.